along the ranges of species across the world, we don't see the diversity below species level in the phenotypes. And this reflects the past climate history and reflects present oceanographic conditions.
Across the world,
and the climate past is imprinted in the present distribution of biodiversity along the species ranges, because especially in areas that have been subjected to very sharp changes in climate in the recent past, such as the higher latitudes, we have this sort of paradigm of climate change where species expand north and south depending on whether we're in the northern hemisphere or southern hemisphere, depending on whether we are in a warming or a cooling period. Presently, we are in a warming period. So if we are in the northern hemisphere, this diagram would represent the present expansion of species towards higher latitudes. But we have to think that in the past, during cooling periods, we had the opposite effect. And so along the present range, along the range of species, there are some areas which have persisted, hopefully, in between the warming and the cooling periods. And these are the ancient populations that are expected to retain most of the diversity along the species ranges, most of the ancient alleles, whereas expansions northwards and southwards and along coastlines normally represent bottlenecks, which are associated with homogeneous diversity and loss of diversity, because fewer alleles are present during these expansion areas. And why is that? So as climate changes, why doesn't all the diversity expand north and south as ranges shift? Well, this diagram represents the expected process. So if you think of a climatic refugia, where conditions start becoming favorable towards some region, the first organism, the first individuals that expand towards some area will have much increased fitness, and they will leave a lot of descendants, they will have no competitors. And so this tends to create these expansion fronts, which are dominated by the offspring of the ones that happen to be at the right place at the right time, and creates these lower diversity homogeneous conditions. So along species ranges, this happens in many coastlines along the world for many species that have been studied, but we don't see this in the phenotypes again. So it is important in terms of planning conservation, planning, even applications such as aquaculture and all types of implications of diversity. It is important to understand where are these ancient populations with private alleles, richer, and there are conservation hotspots that they are very important and often threatened. Often they are located at the rear edge of species ranges where they might be disappearing without being noticed if we don't study them. Okay, this diagram, we're not going into details, is just to remind you that in the recent past, in Northern Europe in particular, in the Northern Hemisphere in general, we have had a lot of variations in sea surface temperatures and climate in general, and relative to the present conditions, we had much cooler periods and some slightly warmer periods in the recent past, and this left marks in the present distribution and structure of marine populations, such as marine forests across the world, and we're going to see some examples of that. So the first example is going to be a kelp forest that occurs presently around the coastlines of Europe. This is the present distribution, but if we use the present conditions under which the species occurs and try to understand if we try to infer where the species might have occurred in the recent coolest period and in the most recent warmest period, we can model these sort of ranges for the recent past. And now we may ask, so what are the populations that haven't suffered bottlenecks haven't suffered extinctions and recolonizations in the recent past. So these are marked here with these squares. These are the sites where we expect to have the ancient populations that didn't go extinct and recolonized due to these climatic changes of the recent past. And if we look at genetic structure of this species along its entire distribution, we find indeed three areas like coincident with this sort of the Celtic Sea spot, the Northwest Iberia, and then some seamounts and upwelling spots. These groups are represented here in these three different colors. And these numbers represent private alleles that are still found in these groups. This is just to highlight that most of the gene pool of the species is found in the southernmost genetic group, which is now restricted to very few regions in the world. It's only present in some upwelling spots of Northwest Africa and Gibraltar and some seamounts. And so most of the gene pool of this species across its entire distribution is restricted to these very few populations. And this is important to understand if we're thinking of conserving these gene pools before they disappear because they are threatened by climate change. Another example, this is the southernmost laminaria species in Europe or in this Eastern Atlantic. And again, we have a similar distribution. However, there's a difference here. So if we take the present distribution and hindcast what might have been its distribution in the recent past, in the coldest and the warmest periods, we find only the only persistent populations were those in Portugal, basically. And then again, seamounts, the Azores, and seamounts and upwelling spots. And if we look at genetic groups, in this case, we have two major genetic groups, and one of them has most of the gene pool of the species again. Most of the private alleles are present in this group, which is only comprising these upwelling spots and some seamounts in the Azores. Most of the species, all the continental distribution of the species in Europe is genetically poor. And this has actually is reflected also in some differences, physiological differences, where northern species, the northern populations, if exposed to heat shock, just never recover, whereas the southern populations, if exposed to heat shock, they recover. Even subsequent heat shocks, they always recover. So there are physiological differences along distribution, even though it's always the same species. If we go somewhere else in the world, to California, and we see the similar pattern, it's the same processes happen, just different species, different coastlines. So for example, this is a major predominant species that occurs along the coast of California, Sylvicia. And if we do again the same exercise of using the present distribution to hindcast the recent past in the coldest and warmest periods, we find that the persistent populations don't extend all the way to the southernmost populations. So the species would have gone extinct in the southernmost populations in the warmest recent periods in the mid-Holocene, and it recently expanded south as well as north. And interestingly, we see this again reflected in the genetic structure of the population. So we find three different genetic groups, one in the colder area, and then two in the warmer area. One is continental and the other one is these islands. And the recent expansion southwards, so the expansion that's predicted by the models, in fact, is also reflected in the genetic structure, in the sense that this is the same genetic group as the islands, indicating that this recent expansion has not been from the continent, but from the islands. Here another example of a different species for the same coastline. In this case, the genetic groups indicate that while the species is almost absent along the coastline, it's only remaining in islands offshore in the southern region. It's on the coastline, it only remains in the northern part. And the southernmost population, we have not found it anymore. So it sounds like it's undergoing extinction. So all these reached southern groups are restricted to very few small habitats, isolated, and some of them might just be disappearing as we watch. Again, for the same coastline of California, if we go to a very charismatic species, Macrocystis purifera, the giant kelp, here again, we see the same type of pattern that, as I mentioned, is a common pattern across the Northern Hemisphere, where we have lower genetic diversity as we go towards north. These yellow spots here are actually Alaska, but they are off the map, so they're plotted in here. And here I just want to highlight again that there are functional differences. These are the results of an experiment done by Luis Barreto and Gareth Pearson. And they just show that if you expose this giant kelp to different temperatures, above a certain temperature, 24 degrees, the northern and north center populations do not recover anymore, but the southern edge in Mexico can still continue to function, and they have different gene expression patterns in different transcriptomes, which we don't get into, in response to this thermal shock that shows these populations, even though it's all the same species, they are physiologically and functionally different. Okay, so continuing now to the southern hemisphere. So we've been looking at the northern hemisphere, which was highly affected by the last glacial maximum, where ice sheets came way south. And so there was this extinction recolonization was very striking, especially in Europe, where all this area was frozen. And the southern hemisphere is a strong contrast. The ice did not cover continents in the same way as in the north. So we have two very contrasting polar zones in our planet. And so if we continue with the same species, giant kelp, which is interestingly occurring both in the northern and the southern hemisphere, we can look at its entire distribution along the Southern Ocean and see that it does contain diversity, and that is present in the Southern Ocean and doesn't show these marks of a strong restriction. And due to extinctions and recolonizations causing loss of alleles. And if we look at the distribution of ice during the last glacial maximum, we can see that there were refugia. For example, here the Falkland Islands was an area that was outside the ice, and it's supposed to be a very important. Our data suggested it was an important refugium for alleles for this giant kelp in the past, and presently. If we look at giant kelp in the Atlantic Ocean, which is present in South Africa, South America, we see that the Falkland Islands are the hotspot where you find the melting point with everything. And here's a special focus on the Falkland Islands, where if you compare specifically several populations from Patagonia, from South Africa, and from the Falklands, we see that the Falklands have mostly an endemic genetic group that's present only there, but also have some from South Africa and Patagonia, they have more diversity, more alleles, and an important spot. Continuing with the Falkland Islands, if we look at another kelp, not giant kelp, but Lawsonian, Southern Ocean marine forests that are very important. They occur in New Zealand, Tasmania, and several islands around the Southern Ocean. And again, here we see that the Falklands are a hotspot of diversity. These areas that were outside the ice, these islands that were outside the ice, in contrast with most of the other Southern Ocean islands, retain unique genetic groups, unique species that we didn't even know there were different species. They're still to be described. We found these distinct genetic groups occurring side by side. So they're obviously different species. They're reproductively isolated in the same island, in the same spot, some segregated by depths, some shallower, some deeper genetic groups. So there's a lot of hidden diversity and hidden speciation processes yet to be discovered in these marine forests of the Southern Ocean. The Arctic is especially interesting because, as I mentioned, the ice sheets came south very strongly, and so isolated the east and western parts of both the Atlantic and in the Pacific also to some extent, although this was more striking in the Atlantic. So the Arctic has functioned as an evolutionary pump where as climate warmed and cooled, it isolated populations between the east and the west of each ocean, and also isolated the oceans because the connection between the Pacific and the Atlantic was possible or not possible, depending on the Bering Strait over there and on the amount of ice here. And this raises several interesting questions. So when we have the same species on both sides of the oceans, for example, on both sides of the Atlantic, did they persist during glacial periods on both sides, or did they persist just on one side and then recolonize the other side? So, for example, there are recent data in collaboration with the team of Roscoff has suggested that this species, this very important kelp species, Laminaria digitata, actually persisted only in the European side and recolonized the American side after the ice receded. But so this ice functions as an extrinsic barrier where populations staying on both sides, when they recontact, they can form just hybrid zones, or if these hybrids have lower fitness, it can even lead to speciation. But let's see if we have examples of that. So in contrast with what I just showed for Laminaria, if we look at Fucus vesiculosus, also a very common species across the Atlantic Ocean, the North Atlantic, we find a different pattern where we have evidence of a distinct genetic group that is ancient in North America and Canada, and versus Europe. In Europe, we have two groups, the southern and the northern one, which are very genetically distinct. And the Arctic, the area that was covered by ice and recolonized, in fact, is presently an admixture between individuals that came from the west and from the east and form these distinct genotypes. In fact, then these are also functionally different, because if we compare their survival and their resistance and resilience to temperature warming, we find when we compare Greenland with Portugal, for example, we see that the Portuguese ones, the southern genotypes, they recover from stress even at 32 degrees, whereas the ones from Greenland don't. So again, it's the same species, but with functional differences across its distribution. And now, interestingly, what can happen is that as species expand, they don't just lose diversity and become homogeneous. Interesting things can happen, such as genome recombinations when they contact other populations or species with which they can hybridize, as we were started to see now with the last example. So if these expansions are taking place during providing opportunities for hybridization, then we can have the introgressed individual spreading, and this can change the species pool throughout entire species ranges throughout very, very large regions. And we can see some evidence for that. The most one very clear evidence already shown many years ago was Fucus serranoides. So this interesting species that's European endemic has its native mitochondria and chloroplasts. These organelles are only native in Northwest Iberia, because all over its distribution, most of its distribution has individuals that have foreign organelles. The mitochondria and chloroplasts of this species north of France actually are from another species. So the hypothesis that during post-glacial recolonization, as it encountered another species introgressed, then it expanded with the introgressed organelles. And as these are threatened by climate, we might be looking at these very ancient native populations disappearing and losing some of this particular diversity that's only restricted now to Northwest Iberia. Some lineages are very, very restricted in some very, very few small areas. So as I was mentioning, when you have this genome recombination, you can form low fitness hybrids, and then they might end up forming intrinsic barriers to reproduction and lead to speciation. We have some examples of that also in the Arctic Ocean. Saccharina latissima is a kelp, sugar kelp is the one with the most, the species with most economic importance perhaps in Europe, because it's nice to eat. It's a nice edible species, widely used in aquaculture. And here's some examples of the way it looks in Alaska, in Portugal and Spain, or in Greenland. So it's the same species all the time, but totally different phenotypes. And if we look at its genetic structure along its entire distribution, which interestingly is across the Arctic to occurring both in the Pacific and in the Atlantic, we have three major groups. So one is the European one, although it's actually comprised by two subgroups, but this is more similar than with the other ones. So the European group, the Canadian Maritimes zone, and then we have the Pacific group that goes from Japan, China to Canada and the United States. Interestingly, in the Arctic, now we start to see areas where we have a mixture of the Pacific and the Atlantic genotypes. And so the question arises, so do they mix or not? What happens when now the Pacific ones seem to be entering the Arctic and meeting the Atlantic types of the same species in Western Greenland and Hudson Bay? What happens there? Well, interestingly, there's very few hybrid, well, we can't call it hybrids because it's the same species, but admixed individuals, there's very few. Most of them are still strictly Pacific or strictly Atlantic. So the individuals sampled in this region, and interestingly, Greenland has European Saccharina latissima on one side and has this admixture on the other side. And when sampled there, most of the individuals sampled are still strictly Pacific or strictly Atlantic. Very few are mixed, suggesting there are some reproductive barriers already evolving. And they're, as I showed in the picture, they are very different morphologically. So you can actually tell them apart. Here's another very interesting case caused by the Arctic speciation pump. Fucus disticus, this is an intertidal species. With very strong genetic structure in the Pacific Ocean and very shallow genetic structure, very homogeneous structure in the Atlantic, indicating that it was, the species has been present in the Pacific for a longer time and recently colonized the Atlantic by crossing the Arctic. Now, if you look at the structure in the Pacific Ocean, which is especially extended along the US coast, the most distinct genetic group is this one marked in red in Southern California. There's a very, very distinct group of this species here. And there are parts, contact zones between this genetic group and the next subsequent one for the north. And where they occur exactly on the same spot, on the same coastline, they do not mix. They're mostly remaining different. Here's actually a picture. They actually look different morphologically. And they have differences and they are not mixing. So they seem to be different species already. And then you have these genetic groups which are endemic to very small stretches of coastline all along the United States until Canada. And the same thing happens in Japan and Russia. When we go to Alaska, we find the melting point. The melting pot. We find that in different places in Alaska, wherever you look, you have a different combination of different genetic groups, of these ones that are so different, so restricted and not mixing along this entire coastline. When you get to Alaska, they all occur mixed up in different combinations, which look random. It looks like this area, which was an unsuitable habitat in the past due to ice, has been recently colonized by different individuals coming from the East and West Pacific. And wherever they occur, they arrive together, they tend to still remain segregated to a certain extent. In many cases, one genetic type is higher than the other one on the shore. And this photograph actually shows that. This is a photograph of Alaska. And this species of fucus, you see here, this band upper on the shore is one of these genetic types. And the other band lower on the shore is the same species, but physiologically distinct because it occurs in different parts of the shore, morphologically distinct, and genetically distinct. So interestingly, after being separated for some period of time, when they actually meet together, there are these almost experimental assemblages of different genetic types mixed up along different spots in Alaska, and they tend to be segregated vertically, physiologically, and genetically distinct. And some admixture is found, but in many cases not really. In many cases they remain different. And here's a better an increased view of Alaska, and these are samples that we have from along the Aleutian Islands all the way to Canada, so along Alaska, and you can see that in different spots have been recolonized from different sources, some from Japan and Russia, some from Canada and America, and again in some cases they are mixing in the same shore level. In many cases they remain separated and segregated vertically. So it's a very interesting experiment that we're following up. Okay, continuing with these processes that you can find when species ranges expand due to climate, and then different recombinations and evolutionary processes are found. One that can happen is that the hybrid individuals become instantaneously reproductively isolated. That's what happens when they become halopolyploid. And here's an example of that. Along between Canada and Mexico, so along the Eastern Pacific, we knew that there were two sister species very closely related in the intertidal zone. This one called Pelvichiopsis limitata, a colder adapted species in the north, and then a more warm adapted species in the south had another name. Now we call it Pelvichiopsis californica. And in the contact zone between the two, we discovered two extra species that were previously unknown. And in this contact zone, these species actually have caused very interesting processes. So one of them, which is the one represented in yellow, Pelvichiopsis arborensis, is one of the most restricted species with the most restricted distribution I know. It only occurs here in the Big Sur and Monterey Bay. And even there, it's only present in very few rocks at the top of the shore. And and then there is another one a little bit further south, which is a hybrid, allopolyploid hybrid. So for every locus, it has one copy from this one, represented in yellow, and another copy from this one, represented in blue. It's an allopolyploid from these two species. Interestingly, the allopolyploid is always occurring above on the shore, above the other species. So they become physiologically differentiated to an extent that they occupy different niches in the habitat. Most unexpectedly, we discovered also that this northern species that has a wide distribution range and is very abundant along the shore is also an allopolyploid hybrid, and its parental species is this tiny one represented in yellow, Pelvichiopsis arborescens, and its maternal species, we never found it. So it seems probably extinct. So the species is extinct, but its genome is still living as half of the genome of Pelvichiopsis limitata. And in fact, the mitochondrial genome of this extinct species is also still alive in this species because the mitochondrial genome is inherited from the mother. So some very interesting processes happening here. Continuing with this group, if we look at expression by these species, well, I won't go into details over here. Just want to say that the expression of the different loci is conserved. So basically the homolog expression match that expression of the parental orthologs. They're not using more the genes from one parental species than the other. And the homologs have increased the non-synonymous divergence versus the parental orthologs. So this suggests that there are some relaxed selective constraints after the allopolyploidization. So perhaps the deleterious substitutions are buffered by having available alleles from the other species. This is just to show the physiological comparisons of these species. If we expose them to higher temperatures, the hybrid withstands even 30 degrees, which are temperatures that none of the parental species can recover from. So neither the maternal or the paternal species withstands temperatures that the hybrids do survive to, whereas the other allopolyploid hybrid is the opposite. It's much less tolerant to warmer temperatures than at least the one parental species that's known. The other one is extinct, of course. So very different, very interesting that these instantaneous genome recombinations result in totally different physiological traits. This is just an example of totally different species, but just to highlight how interesting it is, the hybrid effect in terms of changing the traits of species. So here, this is a study by Neuza Martins, which crossed two different types of kelp, a cold adapted kelp, Laminaria digitata, with Laminaria pallida, with a warm adapted kelp, and the hybrids, no matter which one used as maternal or paternal, in both cases had higher thermal tolerances than both parents, even than the warm tolerant species. At 24, where these hardly survived, they were still surviving and had better survival. And this is just to highlight the warm tolerant species used in those crosses is actually from Namibia and South Africa. It occurs all the way almost to Angola, and has interesting, again, population structure where we find two genetic groups that are very, very distinct, and a contact zone between the two where there's a little admixture. And this is the northern limit of kelps of these kelps in the Atlantic, in the southern hemisphere. This is almost in Angola. This is kelp forests of Laminaria pallida in Namibia. Continuing with the southern hemisphere. So if we go to South Africa now, we have, again, contrasting sister species that have warm and cold adaptations. Here we have Eclonia radiata on the upwelling cold water coast of South Africa, and we have Eclonia maxima on the warm oceanographic conditions of South Africa. There's a gap in between the two species due to unsuitable habitat because it's very sandy and so on. But so what's interesting is that looking at the genetic structure of all these species, which are very, very distinct morphologically, we find that in the contacts, well, not really the contact zone, but in the area in between their distribution, which is where Eclonia maxima has been expanding southwards, there are populations which have the nuclear genome of Eclonia maxima and the mitochondrial genome of Eclonia radiata. Again, in these contact zones between species with contrasting physiological traits can result in different rapid evolution. And these special unique genotypes that are formed perhaps due to hybrid processes actually occur in different habitats. So they are deeper in general below all the other species, or occurring in offshore in seamounts. Okay, so what might happen if climate change continues? So this is the diagram I showed you. As in our case, in the northern hemisphere, if warming continues, we tend to assist species ranges, the edge populations becoming fragmented and smaller, and they might just go extinct completely. So this is what's happening presently in North Atlantic. This is a big concern for Europe. Why? Because for all the species that we looked, basically for marine forests in general, all of the species that we looked, we see that the species are not threatened at all. They have a lot of new habitat opening and expanding into in the northern regions of the Atlantic. But what's threatened is not the species, but the gene pools that are left behind, the rich gene pools that are left behind. And it's just to show you some examples of this vanishing rear edge. And this is a good picture to illustrate how this is possible, because sometimes you might think, oh, how do things go extinct? It's not so easy. But this, for example, this is the entire population of Fucus gyrae, one particular species, in a certain place in the Algarve. This is actually the type population. And that patch is the entire population in this zone here. There's the species has gone extinct in many areas along its distribution. Used to occur all over the Moroccan coast, Northwest Africa. And in this study, our student Carla Lorenzo has gone through the entire coast and found that it only remains in upwelling spots. Upwelling is really important. So these spots allow the persistence of particular genetic groups that have gone extinct in some other places where they had been previously recorded. And in case of the Algarve, just to highlight that these extinctions sometimes recover. And here's a picture of what used to be a luxuriant population of this species, Fucus gyrae, near Albufeira, and then it went extinct for several years, disappeared completely, and now it appeared again. Some individuals, and it's not like that, but it's recovering. And what we see is that it had a unique allele that was not present anywhere else in the world that we know of for this species, except in this population Albufeira. And the ones that are now appearing still have this unique allele. So it's not being recolonized from elsewhere. Somewhere there were some individuals that survived microscopically or something, and the recolonization that's happening now, 10 years later, is still from the same gene pool, which is good news. Here's another example of a massive southern extinction. All these red dots are points where the species was recorded and disappeared. And this was a unique genetic group, so it only remains in very few places, like in the Tagus estuary. And this southern lineage is very threatened. Many other cases exist, and it's here I want to highlight the importance of herbaria, because this species, which presently has the southern limit in northern Portugal, in the 19th century was present south of Lisbon in very large individuals that had been collected by Velvich and are present in the herbarium in Lisbon. And I saw some of it that is now extinct. This is a picture I took in 92, for example. So we're watching these extinctions and this sort of southern contractions towards the north. So extinction of native lineages is happening as we watch. Another case, this one, Bifurcaria bifurcata. This species is very homogeneous throughout its distribution. It's expanding in the British Isles, but most very distinct genetic group occurred in West Africa. It's gone extinct. These populations have not been found anymore that were recorded in Western Sahara. So there's only very few left for this lineage, which I should highlight as most of the diversity of the species. Again, most of the gene pool of the species is in West Africa. What's expanding in the British Isles in the north is a very poor, genetically poor subset of the gene pool of the species. And we should be aware of this. So in this paper, we make a summary of these genetic consequences of climate change. For several kelp species of Europe, these squares represent sort of a refugia where it's expected to have high diversity, the best gene pool, hotspots, and red means these areas are predicted to go extinct. Green, they're predicted to expand. So all the populations are predicted to expand somewhere. The species are not threatened, but the unique gene pools are threatened in several cases. And this is for the major kelp species of Europe, so it's a serious problem. And so we finish with the problem that I started with, which was, okay, there's diversity below species level. This is a picture that illustrates that. This is a marine forest that existed and doesn't exist anymore. We know it existed because someone took a picture of it in 1987, so you can still appreciate how beautiful it used to be. And we can think, so does it matter? The species expanding in Northern Europe, was it different? We know now it was different. We know these populations had most of the gene pool and they're very distinct from the ones expanding in the north. And what can we do about it? And I finish with this question. So there's gene pools going threatened and going extinct. And so if we try to do restoration, now we are in the decade of restoration. A lot of policies recommend restoration. How are we going to recover these populations that had unique gene pools, and now we are not going to recover them with lower diversity than they used to be? How can we save the gene pools? Should we have, and this is a good question for Assemble and for the BRC, should we have important collections to save these gene pools and to use them in restoration practices? Because now we have the information to do that. Now we know, even though we don't see this in the phenotypes, we know what are the populations that have the unique gene pools and the richest hotspots of diversity for the marine forests of Europe. Should we save them? And I finish here with a thank you to lots of people who have been helping this. This is not an extensive list, there's many more. It's just a study that has been possible. All these studies have been possible thanks to a lot of collaborators around the world. Thank you.
Thank you, Ester. So if anyone has any questions, please, you can tell us in the chat so that then you can talk, just so that we just know who is ready to ask questions. Well, I have a question. In fact, so these populations you were presenting are basically, they are essentially fixed on the substrate, so their mobility is relatively reduced, apart from probably the reproductive structures and so on. So how about, what do you think? Do you think there is a similar process going on with, you know, plankton, nekton? You know, do you have any information on this?
Yes, that's a good question. So this is expected mostly for species that have dispersal traits that are very restricted. So there's been a paper by some of our colleagues, for example, for anchovies, where they show that this doesn't happen. They swim, the fish, the adults swim and have larval stages that are abundant and stay a long time in the plankton. So these like small pelagic fish, for example, are not expected to show so much the same trend. In general, the trend, there's not certain species that are doing this and others that are not. You can find examples across a lot of different groups, but they are very strictly consistent in species that have low dispersal. And these would be not just macroalgae, that, well, there are macroalgae that are very dispersive, as you know, from invasive species. So it's not just because they're macroalgae, it's species that have traits that don't allow large scale dispersal, and a lot of invertebrates are even more restricted in terms of dispersal than macroalgae. So benthic invertebrates, from corals and sponges and etc., a lot of them are expected to have even stronger patterns than this. And there are a lot of data and publications that already show that this is the case. So it will depend again on the group. Of course, we don't expect it so much for tuna fish or something like that. But most of the marine species perhaps will show cases like that, because there's a lot of, these traits are very common across a lot of marine species.
Thank you. So Yorgos Kotoulas has a question. You can ask Yorgos.
Yes, this is really great work and congratulations for it. It's quite impressive. And I think it will be useful in many respects. How difficult was it to get access to the material? Was it a major challenge?
Yeah, well, that's also a good question. how to do this. So the best way to do these types of studies is to have a lot of collaborators. There's networks like this through Assemble is very helpful in that respect. We can just contact people that are working in different parts of the region and ask them if they can collaborate and sample, but often it's not easy to sample, or do they recognize the species and so on, but you can always at least try to visit them and go there, collect yourself, because perhaps you need to be an expert to actually find the species and collect them. But the possibility to access different regions is highly facilitated by having networks of collaborators and networks of laboratories and places that you can visit and work with local people. So it's very important, the networking of collaborators and the possibility of different researchers to go to different places. So again, this is to praise Assemble for allowing this.
Okay, so thank you. So now it's Cara Layton. You can ask your question.
Oh, sorry about that. Hi, Esther. Thank you so much. That was really interesting. My question was just that I did join in a little late, so you may have mentioned this, so I apologize. But I was wondering if you did or you would expect to see parallel patterns of loss in species that occur across the Atlantic. So in an amphi-Atlantic species, are you still seeing those same patterns and those range edge effects?
Sorry, I was reading another question in the chat, and your question was not the same one that I was reading. So
okay, no, that's okay.
Can you repeat it?
Of course. Yeah, I was just saying that I was wondering if you have seen or you would expect to see that in amphi-Atlantic species that occur on both sides of the Atlantic Ocean. Are you seeing those same red range edge effects and really those parallel patterns of loss on both sides?
Yeah, well, that's a very interesting question also. Yes, one that comes to my mind immediately is Fucus vesiculosus. We had recorded a huge range loss on the Eastern Atlantic side. And just recently, in work in collaboration with my ex-supervisor, Susan Brawley from Maine, she just recorded the loss of these species across the southern edge from North Carolina and northward. It's the same thing happened on the other side. In fact, we had actually published a model that predicted this loss a few years ago, but we didn't know it was happening. And now, unfortunately, just like predicted by the models, it's happening.
That's sad, but very interesting.
Yeah.
Thank you.
Thank you, Cara. Now, Wiebe Questra, can you ask your question?
Yes. Okay, this is not a question about the population losses and so on, but it is a question about the economics. Laminaria, for instance, or it is now called Saccharina there, is grown in aquaculture in China in huge enterprises, and that is beyond the southern natural limits of the species, because they grow the gametophytes over the summer in cooled greenhouses and then generate the new sporophytes that later in the autumn they put back in the field. So the species would never survive in those warmer areas, but the aquaculture can handle this. Now, it's not a solution to the southern extinction of the species, but it offers economic possibility. So what do you think of this in Europe?
I think this is a great idea. I actually have been mentioning this to some people in the field, suggesting that this is what we should be doing. So in often the conditions in a certain range are ideal most of the year, except for a very short period. In fact, our concern, for example, in Portugal, is often the stormy periods in the winter, and you can just grow things during the favorable season, and then remove them and keep them in culture, and then place again, just like it's done in China. So either to withstand winter storms, or to withstand heat shocks periods, which again happen for very short periods. I think this is an excellent suggestion, and it should be followed, and that would be a leading way to overcome a lot of the bottlenecks that are happening in Europe at the moment for this.
If I add something here, I think one of the problems with aquaculture is, in fact, the very narrow genotypes, because basically you build, it's just a series of bottlenecks. So I think that this is probably a solution where aquaculture may have a limited contribution, I would say, but certainly I would. It's a way. You mentioned before biobanks. I mean, this is essentially. You need a biobank to be able to resolve this. So now we have Lucia, this is the last name here in my chat, Lucia Mascorda. Please, Cabre.
Yes, hi, hello. Thank you, Esther. It was a great presentation. I found it very interesting. So carrying on from this conversation, you mentioned that one of the possibilities that we'd have would be to have gene pools, same as we have like seed banks. And I was wondering if you knew more about this in terms of like how to go about it, like logistics, cost. And obviously you've done all these studies on these species, but we would need far more knowledge on other species to localize where the highest gene pools are in order to save them. So I was wondering if you could expand on this.
Yes, again, great question. I'm glad you asked, because this is the discussion that we need to have, because we know these things these losses are happening. We watch some of them and we predict that some will happen, and some of the ones we predicted are already happening. So it is important to discuss and see if we should do something. The point of saving things as gene pools and seed banks is the scale, right? And the alternative of transplanting things to other regions is altering nature. So seed banks, I think, are very important. Seed banks, in the sense, not really seed banks, but more like artificially saving gene pools as much as possible is always there is this strong problem of scale. We can only preserve certain amounts, which will never represent the entire diversity that exists, but it's much better than not preserving anything and the whole thing going extinct. So I think this is something that we need to pay a lot of attention now, because in 10 years, more things will have disappeared. And how feasible is that? So it depends again on the species as well. Luckily, kelps are very easy to maintain in culture, because you can maintain the gametophytes, the microscopic generation, and you can maintain them in conditions where they survive for years, for decades without growing, but they remain alive. And microalgae actually are amazing in that sense, because we know there are some culture collections started by the classic phycologists of our times that have been there for decades in their collections, and they're still alive. And anytime you need some to do an experiment, you just go and give them the right conditions, and they continue growing again after 40 years in culture. So in a petri dish, kelp forests, right? So kelp forests are great for this, for gene pool seed banks, because you cultivate the gametophytes. So this, I think, we need to go ahead and also give attention to the ones that exist. So start new ones. And we know that some collections exist that are not funded. People don't have the conditions and the technicians and so on to maintain them. And sometimes people retire and throw them away. I know a case like that, for example. I know a great collection in Brazil of Professor Eurico de Oliveira, who retired and threw away his kelp collection. And so this is really a shame. And so that's something that should be done. It's not the only solution, but it really is a good way to start.
So clearly, this is a role of the biological resource centers, which already keep collections and so on. So Nicholas Spade from the MBRC would like to ask a question.
Thank you very much, and thank you, Esther, for a very interesting talk. Following on a bit from the previous colleague's comment on, biobanks and so on, and what can we do? I mean, one thing is storing them, but then we also need to restore them and reintroduce them. And you mentioned in your talk that there've been quite a lot of recommendations made about restoring these kelp forests that we're losing. But my question is really, are there any real policy changes coming from these recommendations? Are they actually being implemented? And finally, are we doing enough to actually highlight the issues coming from this to our decision makers to try and make a real change?
I'm sorry, my internet disconnected me. Are you still there?
I'm still here, yes.
Yeah, okay. Okay, well, I read your question. I didn't hear because I went offline for a while. But anyway, so this was about policies for restoration. Apparently, yes, because now there are funding programs for restoration, and I guess that's a way to make things happen. So if calls open with funding for restoration, that requires European countries to get together, and there's a call open right now this month for European restoration projects, and we're working on that. And so this is a way to make people pay attention to that and start working. So I think it is this is the way to go, is to open calls where groups have to work together. It's a very big advantage of European policy, is to force people to have to work together. Even if they don't know each other, they'll find partners and create a whole that's more than the sum of the parts, right? Instead of making individual, single, small projects. And this is good. And so I think that is going to happen.
Thank you.
Thank you. I don't see any more questions. So if not, thank you very much, Ester. It was a very interesting presentation, and I can see that the audience enjoyed it. So we'll be back at 11:30 European Central Time for a roundtable precisely on the importance of infrastructures for policy. So we'll have a few policymakers to discuss with us, the way forward. Thank you very much, and hope to see you soon. Bye-bye.